Hey everyone, and welcome to RUBIN'S Mind Lab, where we break down the latest research into what it really means. Ever wonder if your brain might be wired a little differently, and what that really means for you? Well, this new study delves into the fascinating world of brain asymmetry and how we can spot unique differences, even in large groups of people. This paper introduces something pretty cool called MRIHeadScan. Think of it as a super smart automated system that looks at brain scans to measure how symmetrical or asymmetrical different parts of the brain are. Now, why is this important? Well, the researchers say that how our brain is shaped, especially how symmetrical or asymmetrical it is, can tell us a lot about how our brain is developing and if there are any problems, but measuring this in lots of people is really tricky. You see, when you look at brain scans from many different people, there are all sorts of challenges. Registration errors, which is basically when the computer struggles to line up all the different brain scans perfectly, can mess things up. Then there is site-induced variance, which means that scans from different hospitals or different machines can look slightly different, and that can hide the real biological signals. And finally, there is something called statistical circularity, which is a bit of a mouthful, but it means you have to be really careful not to accidentally find something just because of how you are looking for it. So the researchers created this automated system, MRIHeadScan, to tackle these problems. They really focused on making their brain scanning alignment process called nonlinear spatial normalization using FNERT. Super robust, they used special ways to start the scanning process to make sure it worked well, even for brains that are quite asymmetrical from the get-go. They tested this on a huge group of over 2,000 people from two big autism research datasets, ABIDI and ABIDI-SIGID, which came from 37 different places. That's a lot of diversity. To deal with those scanner differences, I mentioned they used a clever trick. They looked at the healthy brains and figured out how to adjust all the scans so they were more consistent, without accidentally messing with any real differences that might be present in people with autism. This made the scanner differences much less of a problem, reducing their impact from a big number down to a tiny one. One of the really interesting parts is how they checked if their computer-processed brains matched the actual brain shapes. They compared measurements from the original brain scans with the detailed process scans and found a really strong connection. This means their system accurately captured the brain structure. Now, for the big findings, when they looked at the average brain asymmetry in a big group of people with autism spectrum disorder, ASD, and compared it to a group of typical controls, they didn't find a significant difference. This might sound like a negative result, but it is actually quite important. It suggests that we can't reliably say that people with autism, as a whole group, have a generally more asymmetrical brain shape overall. However, the researchers were more interested in something else, spotting individual differences. They used a method called leave one out cross-validation, which is like taking away one person's scan, figuring out what's unusual about them compared to everyone else, and then doing that for every single person. What they found was really striking. Even though there wasn't a big group difference in overall asymmetry, the group with ASD showed a significantly higher amount of localized structural weirdness or deviations from the norm. This is key. While the average big picture might look similar, when you zoom in on individual brains, people with ASD tend to have more areas that stand out as being a bit different from what's typical. This means that autism might be characterized more by these unique scattered structural differences, rather than a consistent, widespread change in brain shape across everyone with the condition. So, how did they do all this? Let's simplify the methods a bit. First, they made sure the brain scans were good quality. They had a minimum signal quality check, basically making sure the scan wasn't too fuzzy or corrupted. Then comes the really important part, getting the brains lined up. They used a tool called FSL's Nonlinear Registration Tool, or FNERT, for the heavy lifting. But before that, they put in some extra steps to make sure it worked every time, even if a person's head was a bit tilted in the scanner. They used a tool called FSL Reorient 2STD to get the orientation right. And then a clever initial alignment with FLIRT, sometimes using a wide search to ensure it latched onto the correct structure. After the brain is nicely aligned, they segmented it into different tissue types like gray matter, which is where our thinking happens. They then used voxel-based morphometry, or VBM, to look at the volume of gray matter in very small areas called voxels, which are like tiny 3D pixels. To make sure they were measuring true asymmetry and not just quirks of the alignment process, they created a special template by averaging the standard template with its flipped version. This helped prevent the computer from being biased towards one side of the brain during processing. A really clever part of their method is how they dealt with the data from different scanning sites. They used something called statistical harmonization, inspired by a method called COMBAT. They figured out how to adjust the scans based on the healthy brains from each site. Then, they applied those same adjustments to both the ASD and control groups from that site. This way, They could get rid of the scanner differences without accidentally removing any real biological differences related to autism. They also defined a specific way to measure asymmetry called the Asymmetry Index, or AI. It is basically a formula that compares the amount of something in the right side of the brain to the left side. A positive number means the right side is bigger, a negative number means the left side is bigger, and 0 means it's perfectly symmetrical. Now, for finding those individual differences, they calculated z-scores for each tiny part of the brain, comparing it to what is normal in the control group. A z-score tells you how many standard deviations away from the average a particular measurement is. A high z-score means it's quite unusual. They then used a strict method called false discovery rate correction to make sure that the unusual differences they found weren't just random chance. After they identified these unusual spots, they grouped them into clusters. They then measured the mass of these clusters, essentially how much unusualness was in each cluster. This metric, outlier cluster mass, turned out to be the key to finding differences between the groups. The researchers also looked at how successful their main alignment process was. They used a metric called registration success rate. This is a really high success rate, meaning their system is very good at aligning brain scans, even difficult ones. They showed with a figure that their new method worked much better than standard methods for tricky scans. They also confirmed that the detailed VBM measurements were closely related to simpler native space measurements. This is important because it shows that even though they process the brains in a very complex way, they are still capturing fundamental aspects of brain shape that make sense. Let's dig a bit deeper into the results. The fact that they found no significant difference in overall brain asymmetry between the ASD and control groups is actually a strength of their study. It shows that their processing is sensitive enough to not find differences where they don't truly exist on average. However, their refined outlier cluster mass metric, which looks at localized deviations, showed a clear and significant difference. The p-value here, 1.90 times 10 to the power of -5, is incredibly small. That is as close to 0 as you can get for a p-value in practical terms. It means the chance of this result happening just by random luck is astronomically low. The ASD group, on average, had a higher mass of these unusual clusters in their brains. This is where the story gets really interesting. While looking at whole brains doesn't show a consistent difference between groups, looking at individual deviations does. This suggests that the brain differences in autism might be like a unique puzzle for each person. They aren't all following the same pattern on average, but each brain might have its own specific set of unusual parts. This is incredibly important for understanding the diversity within autism. The researchers also checked if these differences were influenced by the scanners. They found a big side bias initially, but their harmonization method completely fixed it. They also looked at whether these brain differences were related to things like age or how severe someone's autism symptoms were. They didn't find strong links between overall brain asymmetry and things like IQ or autism severity scores. This suggests that the structural brain shape differences they found are a bit separate from these behavioral measures, or at least not in a simple, direct way. So, what does this all mean? The MRIHeadScan pipeline seems to be a really powerful tool for looking at brain structure, especially for dealing with the messiness of real-world data from different hospitals and scanners. The big takeaway is that while we might not see a consistent pattern of asymmetry across everyone with autism, there is a real and significant elevation in localized, unique structural anomalies. This points towards a highly individualized neurobiological profile in autism, which has huge implications for how we think about diagnosis and treatment. Instead of looking for one autism brain, we might need to look for each individual's unique brain map. Now, no study is perfect, and the researchers themselves point out some limitations. One is that they had to exclude about 4% of the scans because they didn't meet quality standards. These often come from scans with a lot of movement, which can be common in some individuals with neurological conditions. So, the results might be more representative of those who can remain still during scanning, potentially missing some of the more extreme cases. Also, their study focused only on T1-weighted scans, which give a good look at gray matter shape, but they didn't look at other types of scans that could tell us about white matter or other brain tissues. They also mentioned that comparing their method directly to other popular software packages like FreeSurfer would be useful in the future and getting tests. Retest reliability data for their rarity metrics would be great for ensuring their findings are stable over time. Overall, this is a super exciting development in how we can study the brain. It gives us a more precise way to look for individual differences that might be crucial for understanding complex conditions like autism. It's a step towards a more personalized approach to neuroscience. If you found this breakdown helpful, please give this video a thumbs up, leave a comment below with your thoughts on brain asymmetry. And don't forget to subscribe to RUBIN'S Mind Lab for more explorations into the fascinating world of psychology and neuroscience. Thanks for watching.